Morning again. That Today we are going to continue the nervous system presentations. So this is the second presentation that we have completed the first part last 2 weeks. So here, still some students are coming, right? So today we can start talk about the central nervous system. And as we discussed before, that central nervous system consists of mainly two parts, brain and the spinal cord. So here that I'm going to cover uh, certain structures that are there in the brain. So as you saw in the video, that brain consists of neurons. Right? And we discussed about neurons in details in the previous presentations also. That we said that the neuron has a cell body and uh, the axon and dendrites. So cell body is in gray color and the axons are in white color, so that in the brain that you can see the gray area and the white area. So in addition to this neuron, there are some supporting cells. So these supporting cells are known as neuroglia. So neuroglia cells are somewhat different, actually very different from neuron. So the main difference is that these neuroglial cells are non-excitable cells, because you know that when we talked about the neurons, that we said that it has two main uh, characteristics, that is irritability and the conductivity. So that those neurons get excited and excited to the stimulus coming from either external or internal environment, and then it travels that excitement along the neuron to another neuron, and finally it reaches to the brain. There it will be interpreted, and then the response comes the other way. So, but these neuroglial cells are not like that. They are not excited. So they are not excitable cells, and it makes about quarter to half of the volume of brain tissue. So when you look at the brain, that it is not fully comprised with neurons. So half of the volume maybe consists of these neuroglial cells. And unlike neurons, that neuroglia cells can replicate throughout the life. So there are four types of neuroglia. That is in the central nervous system. So those are asterocytes, oligodendrocytes, microglia, and ependymal cells. Right? There are four types: asterocytes, oligodendrocytes, microglia, and ependymal cells. So let's have a look at the first type of cells, asterocytes. So asterocytes are the main supporting tissues of the central nervous system, and its shape is star-like shape with fine branching processes. Right? You know that star has a body, and then the periphery is thin. So similar shape, and it has fine branching processes. Then these foot processes are found in some very small swelling. So if you look at this picture here, so these are the the end of the processes, and you can see that there's a foot process, that there is a little enlargement in swelling, right? So here it is small, and here you can see the process has been swelled up. So so why that it is like that? Because it formed a sleeve, right, around the blood vessel. So sleeve means a covering, right? So just like if I show you my shirt, this is a sleeve, right? Like it covers the whole arm. So similar covering is there. It covers around the blood vessel. So and covering the blood vessel form a structure called blood-brain barrier. So what the importance of blood-brain barrier is that it prevents a toxic substance to enter to the brain. So here the blood vessels, and blood vessel carries a various substance other than the blood cells. There can be some chemicals uh, travels in the plasma. So those toxic chemicals, if those toxic chemicals enters to the brain, that can harm the neurons. So to prevent that, this asterocytes covers the capillary, and those substance that supply to the neurons should pass through the cell wall and also through the food processes, so that it selects the substance that comes out from the blood vessels. So that prevents harmful substance enter to the brain, because the brain is very important structure in the brain, the main controlling system of the body, and also it's a highly sensitive structure. Therefore, that natural method has been designed to prevent those toxic materials like medicine, then the alcohol, that kind of things that people consume will not reach fully to the brain. When neuron damage, phagocytosed and fill the neuroglia cell. So as you know that when a neuron is damaged, that we expect there's no recovery. So in such case, that those damaged neurons will be phagocytosed, that is, it will be cut into pieces and absorbed by the neuroglial cells, and then that gap which created from the damaged neurons will be filled with these neuroglial cells. So in this diagram that you can see how it happens. Now here is a blood vessels, and the vessel wall is covered with the asterocytes and feeds, right? And then the nutritions come uh, into the brain. It will be supplied to the neuron. Right? So here a kind of a synapse, right? Modulation of communication, and here it's supporting coverage to neuron, and this is a myelin sheath, so on. And here nutrient transported from blood to neuron, and this is the asterocytes, which is a star-shaped supporting cell in the nervous system. And the next one is oligodendrocytes. So oligodendrocyte is also similar to the similar in shape to asterocytes, but small in size and also fewer processes. Then asterocytes form and maintain myelin. Asterocytes, now you can see that it formed myelin, and it maintained the amount of myelin. Then oligodendrocytes are found in cluster around nerve cells in gray matter, and also adjacent to the adjacent and around the myelinated nerve fibers. So here a diagram of oligodendrocytes. You can see that oligodendrocytes also covers the myelin sheath. Then the next one is microglia. Microglia are the small amoebial and phagocytic cells. So this is a kind of phagocytic cell. And microglia are scattered throughout the central nervous system, but more in gray matter. Microglia accounts for 10 to 15% of all cells found within the brain. So that the amount of microglia is not that much compared to astrocytes and oligodendrocytes, because it's about 10 to 15% of all cells found in the brain. And it derives from monocytes that migrate from blood. You must be knowing that monocytes are a kind of cells found in the blood. So those cells are help to protect the body from microorganisms. So then, so those monocytes derived and migrate to the brain, and it convert to microglia. Cell become enlarged in areas of inflammation and cell destruction. So these cells, these microglial cells, active when there's an inflammation and cell destruction is taken place. So the main function of the microglial cell is to attack those microorganisms and destroy them. In this diagram, you can see that microglia, this is a resting microglia. It has process of microglial cells and the cell body. And when there's when there is a microorganism, that it activate the microglia, and you can see that the size become bigger, and then it attacks the microorganism, and the microorganism is phagocytosed, that is, it engulf and destroy, right? So it prevent this neurotoxicity, and it gives a kind of neuroprotection, right? So the neurons are protected from microorganisms by microglia. Then the last supporting type of cell is ependymal cell. Ependymal cells lie within the ventricles. So I hope that you heard about ventricles when the video shows the deep structures of the brain. It shows some hollows or open areas within the brain, right? Some cavities, and the presenter said those are ventricles, right? She talks about two lateral ventricles, then the third ventricle and fourth ventricle, which were in the central of the cerebrum, of the brain, rather. So these ependymal cells are the lining of the ventricles, right? And it is comprised with columnar epithelial cells, and this outer surface of the epithelial cells has fine hairs, right, which we call cilia. So cilia is found in the free surface, and this cilia, this fine filament like structure, helps to flow the CSF. CSF means cerebrospinal fluid, right? So that the ventricles are filled with the cerebrospinal fluid, and the cerebrospinal fluid is travels in the ventricular system, and this ependymal cells support that traveling through the movements of the cilia. In this picture, you can see the ependymal cells, which is the lining cell of the ventricle, and it is a columnar epithelial cells. You know, it looks like a column, right? You know what column is, that even in a building, that those pillars coming from the ground to the roof, right? Those are called columns, right? So those are like tall square-shaped structures. So here also you can see that this particular cell is fairly tall, right? This is the nucleus, and on the free surface, this area, this free surface open to the ventricular cavity, and the surface is comprised of small projections, right, like hair, which is called cilia, and this cilia helps to transport cerebrospinal fluid. So here, in general, the functions of the glial cells and ependymal cells. So this is a general statement, general thing, that considering all four types of neuroglia. So one is supporting component of the nervous system, as we heard from the first slide, that neuroglia are the supporting cells of the nervous system, and it acts as an insulator, right? Because it protects the inner structures and act as a defense mechanism by phagocytosis of foreign material and provide limited repair. So microglia helps to engulf and destroy those microorganisms, right? So that is called phagocytosis action. Then oligodendrocytes maintain the myelin sheath, and ependymal cells are associated with cerebrospinal fluid. So this is a kind of question that can come at the exam even. You will be asked to write the functions of each and every neuroglial cells, or in general the functions of the neuroglia, so that you should be able to know, right, what are those functions. So here you can see the types of neuroglia as we discussed before. Now discuss about these four types: asterocytes, oligodendrocytes, microglia, and ependymal cells. And in the peripheral nervous system, there are two kinds of neuroglia. One is called satellite cell. The other one is called Schwann cells. Right. So you heard about Schwann cells a lot when we have our first presentation. So these are the main neuroglial cells, and in the central nervous system, and these two are the main neuroglial, or the protecting cells of the peripheral nervous system. So with that note, I will wind up this session, because the time is going to be ended up. And please connect again, and then we'll move to the next presentation. Thank you. So as we said, that brain consists of neurons and the supporting cells called neuroglia. And we discuss about four types of neuroglia, that is asterocytes, oligodendrocytes, microglia, and ependymal cells. So when we talked about ependymal cells, I said that it is a column epithelial cells which contains cilia at the free surface, and it is the lining of the structure called ventricles in the brain, and this ventricle is filled with spinal fluid, and this microglia helps to the movement of cerebrospinal fluid. So let's see what these ventricles that are found in the central nervous system. So there are four irregular shaped cavities located within the brain. You have seen that in the videos, and it contains fluid, a clear fluid called cerebrospinal fluid. So these four irregular shaped cavities are two lateral ventricles, third ventricle, and fourth ventricle. So you will realize that these irregular shaped cavities are named. So as two lateral ventricles. Right? So the first lateral ventricle and the second lateral ventricle. Then you have the third ventricle and the fourth ventricle. So you saw a 3D diagram of these ventricles in the video. However, when you look at this diagram, you can find out where the ventricles are. And in this diagram, these ventricles are drawn or colored in this light blue color, right? So here, let's see what the structures are. Here you can see that a C-shaped structure located deep inside the cerebral hemispheres, right? And so these two are the lateral ventricles, and the lateral ventricles has a specific shape, right? You can see. So this is anterior-posterior view, right? And this is so here the anterior pole, anterior end, the posterior side. So here you can see that this structure is the lateral ventricle. This is transverse, like what you can see here. So this is the anterior horn. Horn, you know the meaning of horn. Can somebody tell me what is the meaning of horn? What is a horn?
A protrusion, sir?
Yes, that's right. So, like what you see in the bulls, right? And bears, those are again angahela, right? And it looks like that, right? A kind of protrusion. So, that there is a protrusion in the anterior side, so that is called anterior horn, and angavagekak, posterior side, it is called posterior horn, and then to the lateral side, right? And that one is called inferior horn because it goes downwards. So this each lateral ventricle consists of anterior horn, posterior horn, and inferior horn. And these two lateral ventricles, see here the other one, two lateral ventricles are connected, and then here inside is the third ventricle, and the lateral ventricle and the third ventricle is also connected, and then the third ventricle and downwards here is the fourth ventricle. So the third ventricle and the fourth ventricle are also connected, right? And those names are given. So this is anterior horn, this is interventricular foramen. connects lateral ventricles and third ventricle. So as I said that this is the third ventricle with the lateral ventricle, and both lateral ventricles are connected to the third ventricle. The third ventricle is located in the middle, right? In the middle or the mid sagittal area. So therefore that you can see only one lateral ventricle. One third ventricle and one fourth ventricle. Okay? Because it must be the two halves get fused and it looks like one structure, right? So this third ventricle, these two lateral ventricles are connected at the midline, and then there's an opening to the third ventricle. That particular opening is called interventricular foramen. Right? Connects lateral ventricles to third ventricle. So this is interventricular, in between the ventricles, right? Ventricles අතර තියෙන foramen, procedura, which connects the lateral ventricles and third ventricle. Then here the fourth ventricle, right? Here the fourth ventricle. Fourth ventricle is bit enlarged. They call it lozenge shape. And this fourth ventricle is connected to third ventricle via a small duct, right? Duct, yani nalayak, nalayak. So this third ventricle connected to the fourth ventricle by a duct, which is called cerebral aqueduct. So a cerebral aqueduct connect third and fourth ventricles, right? Then this fourth ventricle doesn't ends here. Right? It continue along the spinal cord. So in that area, it is, you know, the spinal cord is not it is a continuation of the brain. But when we talked about central nervous system, we talked brain and spinal cord separately. So therefore here that we talked about the brain. So that I'm not going to talk about spinal cord at this lecture. So, but here you need to understand that in the spinal cord, it continues as central canal, right? As central canal. So the fluid with the fourth ventricle passes down along the. So as we discussed about the ventricular system, we have two lateral ventricles, third ventricle and the fourth ventricle, and two lateral ventricles connect to the third ventricle by interventricular foramen, and third to the fourth by cerebral aqueduct. Then the fourth ventricle travels continue with the central canal of the spinal cord. In addition to that, this fourth ventricle connects to the space called subarachnoid space of the meninges, which will be discussed later, and it connects to the subarachnoid space through two lateral apertures. You can see that two sides of the fourth ventricle, and also a thing called median aperture, right? One median aperture and two lateral apertures. With that, it connects to the subarachnoid space, and as a result of that, the cerebrospinal fluid travels along the ventricular system and also to the subarachnoid space. So in this diagram also you can have the positions of the ventricle. The ventricles are in yellow color, so which is very similar to what you have shown in the previous slide. But here you can get an idea where the brain is located. So as you saw in the video that this big part is cerebrum, and behind the cerebrum is the cerebellum, and here this is the brain stem, right? Stem කියන්නේ නට්ට කියන idea, correct? නට්ට මල්වල තියෙන stem එක. So here the brain stem consists of three areas that is called midbrain, pons, and medulla. Then it continues, it comes out from the foramen magnum of the skull, if you remember, and it continues downwards as the spinal cord. So about lateral ventricle lie within the cerebral hemispheres. Right? This is the two cerebral hemispheres, and one in each side just below the corpus callosum. Remember that in the video they talked about a structure called corpus callosum. Corpus callosum are the white matter, are the fibers which connects these two sides of the brain. Right? And this corpus callosum passes here, right, in between the two cerebral hemispheres. So that is lateral ventricle is located behind, right, or below, rather not behind but below the corpus callosum, right, and separated. These two lateral ventricles are separated. It's separated by a thin membrane called septum lucidum. Right? This thin membrane is called septum lucidum, and the lateral ventricles are lined. That is, the wall is formed by a cilial epithelium, which is called ependymal cells, as you learn. Then connects to the third ventricles. At these two lateral ventricles connect to the third ventricle by intraventricular foramina, right? By opening, which is called intraventricular foramina. Then about the third ventricle, it is a cavity situated below the lateral ventricles between two parts of thalamus, right? And communicate with the fourth ventricle by a canal called cerebral aqueduct. So here is the third ventricle. And either side of the third ventricle, here there is a gray area. You know that gray means it's a collection of nerve cells. So this collection of nerve cells, right, which is around, which is very close to third ventricle, is the thalamus, right? And so that the lateral ventricle is located next to the thalamus, and then it continues downwards to the fourth ventricle by connected by a fairly long canal, right? A fairly long tube. So that is called cerebral aqueduct. Right? So communicated fourth ventricle by canal called cerebral aqueduct. And here also you can see that cerebral aqueduct fairly long. Then the fourth ventricle, it says that fourth ventricle is lozenge shaped cavity, right? It's a shape of a lozenge, right? I don't know whether you have had you eat lozenges as nowadays, because when we were small, that lozenges are very common. Now also that certain like these hacks, right, which helps to soothe the throat. You know, it comes in lozenge shapes. So lozenge shape is that it's a square shape or diamond shape thing, right? Situated behind and below the third ventricle between cerebellum. Let's continue. As I said that the fourth ventricle is lozenge shaped cavity, and it's situated behind and below the third ventricle, and third ventricle between cerebellum and pons. So I said that the lateral ventricles are situated below the corpus callosum. Third ventricle is located either side of the thalamus, and the fourth ventricle is located uh, be, uh, is located behind and below. Sorry, behind the uh, behind and below the third ventricle and between cerebellum and the pons. Right? Pons is in the anterior side and cerebellum is in the posterior side. So the fourth ventricle continue below with the central canal of the spinal cord, and fourth ventricle communicate with the subarachnoid space by a foramina in its roof. I told you that there are two lateral apertures. Apertures is openings, lateral side of the fourth ventricle, and also one median aperture at the roof of the fourth ventricle. So let's have a look at those foramina of the ventricular system. Right, those are the openings of the ventricles to other ventricles. So the first one is interventricular foramina, or we call it for Monro, and it connects the lateral ventricles to the third ventricle. Right, this should be third ventricle. There are no third ventricles. Cerebral aqueduct connects third ventricle to the fourth ventricle. Median aperture or Magendie, it connects fourth ventricle to the subarachnoid space, and then right and left lateral apertures, right? That is right side of the lateral side, and also left side of the lateral side, right? So that is both sides, right? Right side of the lateral side and the left side of the lateral side. It connects subarachnoid space via cistern of the great cerebral vein. So it fourth ventricle connect to the subarachnoid space. So as I said before, that this ventricular system these cavities consist of a fluid. Right? That fluid is called cerebrospinal fluid. So cerebrospinal fluid is secreted to each ventricle by a place called choroid plexus, right? That is the lining membrane of the ventricle walls, and profusion of blood capillaries. So in the ventricular walls, there's a place called choroid plexus, and from that this actually the blood plasma convert and form the cerebrospinal fluid, and then it secreted to the ventricles. Then this cerebrospinal fluid travels along the ventricular system, and finally it passes back to blood through tiny diverticula or diverticula of the arachnoid matter called arachnoid villi, right? Or arachnoid granulations project into the venous sinuses. So I told you that these ventricles connected to the subarachnoid space, right, from the fourth ventricle through the two lateral apertures and one median aperture. So then the cerebrospinal fluid produced in the ventricles passes to the subarachnoid space, and in the subarachnoid space there is a structure called arachnoid villi, right, or arachnoid granulations, which absorb the cerebrospinal fluid, and it is taken to the venous sinuses and to the venous blood, and carried away from the ventricular system. Then the movement of the CSF depends on the difference of the pressure, right? No intrinsic system. So that this cerebrospinal fluid actually circulate, right? But it circulated in the system by itself. It doesn't have a pump to pressurized and supply. Now, when you look at the cardiovascular system, right, heart is the pump. That heart contract and relax at a certain speed, so that when the heart muscles get contracted, especially the ventricles, it pump the blood to the body. But in the cerebrospinal fluid, this system, there is no such pumping action, right? But the movement depends on the difference of the pressure, right? Different different of difference of the pressure in the body. So things like, when you are coughing, when you are sneezing, right, that increase the pressure, and it helps to travel the cerebrospinal fluid. So about the constitution of the cerebrospinal fluid, it is a clear fluid like uh, it is a clear fluid, right? Like water. It's very clear, but yeah, it is almost like water, but slightly alkaline. It is not acidic; it's alkaline. So that means pH is little more than 7. Specific gravity is about 1.007, right? So you know that the specific gravity of the water is 1. So this is 1.007. So very, very similar to that of water. And it contains proteins at about 15 to 18 milligram uh, per liter, I suppose. Then glucose. So that a cerebrospinal fluid is a clear fluid, just like water, slightly alkaline, and the specific gravity is about 1.007, and it contains protein, glucose, calcium, potassium, sodium chloride, bicarbonate, and inorganic phosphates. Right? So you can see that those are in very tiny amounts, right? And that this cerebrospinal fluid has a pressure, right? A certain amount of pressure. So it is about 80 to 100 millimeters of hydro, right? Or 4.4 to 7.3 millimeters of mercury. So you can see that the CSF pressure generated in the brain is very less compared to the blood pressure, right? Blood pressure is 80 to 120 uh, uh, millimeters of mercury, that is systolic and diastolic and systolic pressure. So here, yeah, this is not like that, but it will be in between 4.4 to 7.3 millimeters of mercury. It changes in coughing, sneezing, straining, or lifting weight, right? So when you lift something, when you are straining, right, this pressure will be changed. And we have about 130 milliliters of cerebrospinal fluid. So I told you that cerebrospinal fluid is secreted from the brain, from the lateral ventricles, right? And it secretes continuously at a rate about 0.5 ml milliliters per minute. So that means about 720 milliliters per day, right? So always, every time that the cerebrospinal fluid is secreted to the system, so that if you collected them, it will be about 720 milliliters per day. So you know that the amount of cerebrospinal fluid is found in the body at one point is 130 ml. Therefore, that the CSF will be refreshed, right, about 3 times or 4 times a day, right? So you can see that it's a very clear and fresh fluid. And the function of the cerebrospinal fluid is protect the very delicate nervous system, and it maintain uniform pressure around the structure, right? And it helps for the shock absorbing effect, and it keep brain and spinal cord moist. All substance to interchange between fluids and brain cells, and cerebrospinal fluid gives a buoyancy to the brain and spinal cord so that feel only 1/3 of the weight of the brain, right? So normally that brain weight is about 1,400 grams, right? It's like about 1.5 kilogram, and but we don't feel the weight of the brain because uh, it is uh, filled with this cerebrospinal fluid, right? So you can see the functions of the cerebrospinal fluid, that it protect very delicate nervous system, maintain uniform pressure around the structure. That is very important. If one place get pressed, then that ventricle also get pressed and try to maintain the same pressure, right? So it makes sure there's uniform pressure in the brain. And then shock absorbing effect. So you know that when there are fluid filled sacs are located within the brain, if there is a shock, that it can be absorbed by these sacs, and then keep the brain and the spinal cord moist. So because this is all filled with fluid, therefore that brain is not a dry structure, right? It's a wet structure, and that wetness can be maintained by having cerebrospinal fluid. And then all substance to interchange between fluid and brain cells. So this has given a media or a medium to transport substances, right, from brain cells to the to excrete or to get the substance from the cerebrospinal fluid and pass it to the cells, and also the harmful substance from the neurons to the fluid. And it gives buoyancy, as I said, that it gives a kind of right? It helps to reduce the feeling of weight of the brain and also the spinal cord. Then the conditions following dysfunctions of the ventricular system. So why that you need to learn about this ventricular system sometime, that it can cause various problems, right? So one is that obstructive hydrocephalus, enlargement of head due to accumulation of CSF in the brain. So perhaps, no, you haven't seen patients yet. Even in the orientation visit, I don't know whether you have paid attention to the patients that you have seen. But when you go to Lady Ridgey Hospital, especially in pediatrics places, that you can sometimes see that children with a swollen head, right? The head is bigger than the body. So that is because that due to some reason, the cerebrospinal fluid circulation get blocked, right? So when it is get blocked in the immature brain, right, and the immature that is in the early stages in the pregnancy and the infancy, that you must have noticed that our skull bone is fused, right? We don't have one big skull bone, but we have several skull bones got together, right? You know that frontal bone and two parietal bones and two temporal and one occipital bones, right? So these bones are interconnected by sutures. these places are immature and there is a possibility to expand, when they are in the womb and also in the infancy period. So when there is a CSF collected in the brain, that it gives rise a pressure, right? So that the skull bones become enlarged. As a result of that, their head gets swollen, and this expansion has a limit, right? That the brain will not, the skull will not burst, and it will extend it to the maximum. After that, the brain matter will get pressed, right? All these neurons and the neuroglia get pressed to the skull bone, so that these children will ended up with various disabilities, physical and intellectual disabilities, so that they will come to therapy, right? So such things can be seen in your clinical practice. Therefore, it is important for you to have an idea about this ventricular system. And the treatment is extraventricular drain. So if a child comes with the obstructive hydrocephalus, then there's a surgery. The child will undergo a surgery, and extraventricular drain is inserted. So there's another word called that VP shunt, right? Is inserted. That is, VP means ventricular peritoneal shunt, right? That is an external circuit, like what you see in this picture. So here in this diagram, you can see that in the ventricles, and due to some reasons that this cerebrospinal fluid will not be absorbed to the arachnoid villi and convert its ends to the venous drainage. So in such case, that a ventricular catheter is used, right? Catheter is a kind of a tube insert to the body to a certain gland, or like a place like bladder, to drain the fluid, right? So that a catheter is inserted, and then a catheter lies tunneled under the skin, right? It brings in the neck region under the skin. So sometimes you can see the tube. You can feel the tube. You can see the elevation of the skin, and then it ends. Tube empties into the chest or abdominal cavity, right? And there the fluid will be absorbed. So it is kept in such a way that after a certain pressure, mount in the ventricles, that will send that excess of fluid through this new system, through the VP shunt, to uh, the chest area, away from the brain, so which can prevent increasing unwanted pressure in the brain, and that can prevent brain damages. So when the cerebrospinal fluid um, get accumulated, the pressure will be mounted. So as a result of that, the person will complain of various things, like the person will complain severe headache, or sometimes that severe neck pain, like neck gets stiff and the back gets stiff back pain. So in such case, that doctors wanted to find out whether the cerebrospinal fluid is blocked, and why that it is blocked, whether it is a natural reason or whether it is an acquired reason, like whether there's an infection, enters to the cerebrospinal fluid and block the pathway, or whether there's a tumor, right, or a cancer grown up in the ventricular system and it block the circulation, so on. So therefore, that cerebrospinal fluid is taken out by using a needle, which we call lumbar puncture. Right? So you know lumbar is the lumbar region. Puncture means Sidurakudina, right? Creating a little hole. So CSF pressure is measured using tube attached to lumbar puncture needle. The brain intracranial pressure rise due to hemorrhage or tumor. Compensation is made by a reduction in the amount of CSF. Yeah, so as I said that it can happen due to infection, enters to the cerebrospinal fluid, and also if something goes wrong in the brain, let's say that a tumor is started mounting and developing, so that uh, it press the ventricles, and then the pressure in the ventricles will go up, right? So that it can be measured by doing a lumbar puncture. If volume of the brain matter reduces, the volume of the CSF increases. So that you will perhaps see one day when you are doing clinical practice that a needle is sent to the lumbar region, specifically between L3 and L4 vertebra, and you know here the spinal cord is not found because spinal cord ends at L1 and L2, and these are a set of nerve fibers, and this needle doesn't hit on the nerve fibers, it goes only to the space, and it says cerebrospinal fluid collected from the thecal sac that surrounds the spinal cord, and then when this cap is removed, that cerebrospinal fluid will start flowing down, and they have a meter to check the pressure, and then when it's open up, that the fluid start falling down with like a dot by dot, and it is collected to a test tube, and then it is sent for testing. So in that way we can find out whether whether there is a infection or whether there is any other issue, whether there's an abnormality in the cerebrospinal fluid that will help to diagnose the problem, right? So this is all about cerebrospinal fluid and the ventricular system. So we will continue uh, through the nervous system in the next class, right? So do you have any questions or any clarifications? It's the time for discussion.